And one more organelle. This last organelle is the chloroplast, and you do not find chloroplasts in animal cells. You find them in plant cells and some protistans. Remember, protistans are typically single-celled eukaryotes. Like if you looked at pond scum under a microscope, you would see some protistans. Anyway, in plant cells and some protistans, you find these structures called chloroplasts, and they are a triple membrane organelle. It's getting more and more crazy. They have 3 phospholipid bilayers. If we zoom in on one, you can see it right there. In chloroplasts, the 2 outermost membranes are smooth, but the innermost membrane is highly folded. It almost looks like it's folded into stacks of coins or pancakes or something. And the chloroplasts are where photosynthesis takes place. When sunlight shines on the plant or the protist, whatever it is we're talking about that has chloroplasts, using the energy of the sunlight, the chloroplast is able to manufacture glucose. Like so. Now, it'd be kind of nice if we had chloroplast, then you can just get in the light like this and you wouldn't have to eat lunch. You could say, oh, great glucose I'm making for myself. But unfortunately, we're not able to do that. Only plants and other protistans can. All right, let's take a moment to talk about some organelles that plant cells have and animal cells don't. This is a good review spot. Well, I guess we just covered the chloroplasts. Plant cells have those, but animal cells don't. What other organelle do plant cells have and animal cells do not? So what?
Cell wall.
So good, excellent. I wasn't sure if anybody was going to remember that from last week. That's absolutely correct. Plant cells have a cell wall that's made out of cellulose that's outside their plasma membrane just to protect the cell. It makes a nice rigid cage to protect the cell, whereas animal cells don't have any cell wall. And what else do plant cells have that we don't? Central vacuole, that's right. We have vacuoles, like there's a vacuole and there's a vacuole and there's a vacuole, but we don't have this gigantic central vacuole that pushes everything else off to the side of the cell. All right, that's it for the membranous organelles. And remember about the membranous organelles that eukaryotes have them and prokaryotes don't. Now let's talk about the non-membranous organelles, which are the ones that they're still organelles, they're still structures inside the cells, but they're not made out of membrane. And so prokaryotes can and do have some of these non-membranous organelles we're about to cover, as do eukaryotic cells. Let's start off with the cytoskeleton. Here in your notes is a list of the 3 non-membranous organelles I want to talk about. The cytoskeleton, the centrioles, and the ribosome. If you look at a cell under a microscope like this one, you can see certain structures. I mean, there's the nucleus, there's the plasma membrane, and this grainy looking appearance stuff here are some of the other organelles, which I guess would include some of the endoplasmic reticulum, and I can't really see them clearly, but the Golgi is probably there, and some mitochondria, and vesicles, and lysosomes, and things. What some of those dots are. But you can't really see the cytoskeleton inside the cell, but with a light microscope. But if you first stain the cell with some special stains for detecting the cytoskeleton, then it becomes very apparent. This is a cell pretty similar to the last one we saw, where they have stained it with some of these special stains for the cytoskeleton, and you can see there's a whole bunch of those cytoskeletal structures in there. So what is it? Well, it's a system of protein fibers, of fibrous proteins all linked together that forms a network throughout the entire cytoplasm of the cell. And its name says a lot. Cyto means cell and skeleton means bones, and so it's very much like the bones of the cell. The bones of your body give your body its shape and hold your organs in place. That's pretty much what the cytoskeleton does. It controls the shape of the cell, it gives the cell its shape, and it holds the organelles in their correct places inside the cell. Here's my equivalent cartoon. There's the cell as we viewed it so far, just focusing in on the membranous organelles. But if you could see the cytoskeleton, it would look something like this. It's these fibrous proteins running here and running there and crisscrossing with each other throughout the entire cytoplasm of the cell. And then notice in this cartoon I used two different types of colors for the cytoskeletal proteins, some green ones and some red ones, because there are several different types of cytoskeletal proteins, but the two major types are called microtubules, which I'm going to be showing in green, and actin filaments that I'm going to be showing in red. Okay, what do they do? Well, I guess I mentioned this. They hold the organelles in place. They make like a rigid framework inside the cell that keeps the organelles more or less in one location throughout the cell. And they also control the shape of the cell. If you think about it, the shape of your hand is governed because the bones inside there have those particular shape. Or the shape of your skull is the shape it is because of the skull bones inside that give it its shape. Well, the same with the cell. If the cell has kind of a round shape like this, it's because its cytoskeleton proteins are arranged in a more or less a ball shape. Some cells have these slightly different looking shapes, like the one on the top, for instance, is a muscle cell. It's shaped like a long cigar with rounded ends, and the one below there is a nerve cell, which has an even bizarrer shape, almost like a long elephant trunk, and there's the elephant head over there. But again, what gives a cell its shape is its cytoskeleton. If you look inside nerve cells or muscle cells, you see these cytoskeletal proteins arranged in shapes that give those cells their particular shapes. And a really good example of that are structures that stick out of the cell called cilia and flagella that are used for the cell for propulsion. And let's talk about those, but what we're going to see is, again, it's cytoskeletal proteins that give those cilia and flagella their structure. So some cells have ways of propelling themselves around, swimming around. And I guess the one on top is supposed to be a sperm cell, and it has this very long structure here called the flagella, which is kind of beats like that, and that propels it along. And other cells, instead of having one long whip-like structure, have lots of tiny little hair-like structures those are called cilia. But the principle is the same. The cell will wiggle those, will beat those, and that will propel it along. A little bit later on in the semester in the laboratory, you'll look at some cells like this called paramecium that you'll watch them swimming around under the microscope as they beat their little cilia. Well, the point I'm making here is that what gives these structures, the cilia and the flagella, their shape, are the cytoskeletal proteins. If you could look at the proteins inside there, you would see that it was actually microtubules, one of the cytoskeletal protein types that you always find inside cilia and flagella. Now you don't have to know this, but in case you're curious, each cilia and each flagella has 9 microtubules inside of it. I'm not sure why Mother Nature arrived at that particular number, but that is always the internal structure of all cilia and all flagella is 9 microtubules. Alrighty. Well, in addition to giving the cell its shape and holding the organelles in place, the cytoskeleton has one more function. It serves as railroad tracks for moving the vesicles around. Oh, I think I misspelled vesicle there, it looks like. Well, it's supposed to say vesicle. So remember, the vesicles are those little tiny membranous spheres that carry stuff from one organelle to another. You might have wondered, matter of fact, I think somebody did wonder when I showed this earlier, how the vesicles know where they're supposed to go. Like when a vesicle is launched from the smooth ER, how does it know to drift over to the Golgi body? Well, the answer is the vesicles use the cytoskeletal proteins as railroad tracks. The vesicles know if I go along this cytoskeletal protein, it leads to the Golgi, and so that's where I'm trying to get. That's the track I should take. Or the ones that get launched out of the Golgi, if they're supposed to go to the membrane, they know to take this cytoskeletal protein and then turn on that cytoskeletal protein to get to their destination. I remember reading a little bit more about this, and it's a fascinating subject. What propels the vesicles along those cytoskeletal tracks are actual little proteins called motor proteins. These are enzymes that essentially crawl along the cytoskeletal protein, like a rock climber climbing up a rope. The motor proteins use ATP as their power source, but they crawl along the cytoskeletal proteins, but they drag along vesicles as they go. So those motor proteins are the engines for the freight trains of vesicles, you can think of them as. Cells are amazing things. It is almost like a little complex city inside each and every little cell. Well, let's take one more close look at these two. So, as I said, there are several different varieties of cytoskeletal proteins, but the two major ones are called microtubules and actin filaments. Here's a slide comparing them. Both types are actually made out of smaller proteins that are polymerized together, which is kind of a neat concept in and of itself, because remember, proteins are made out of amino acids linked together, and so now we are seeing proteins themselves acting as polymers to be linked together into larger units. Microtubules, which are shown here, are hollow tubes that are made out of these smaller proteins forming the walls of the tube, like that little cardboard tube in the middle of your toilet paper thing.